Hello folks, this is a short video on questions on photosynthesis. The video is really for the benefit of people who are doing tuition with me. People who are doing tuition with me will already have a full set of my notes and a set of questions, past paper questions on the photosynthesis topic. They'll also have all their ancillary questions as well, so they'll have a complete package of stuff. And really, you can only benefit from this video properly if you do have a set of these questions in front of you here, because I'll be referring to these questions. I'm going through them now over the next 45 minutes or so. Okay, so if you have the question booklet in front of you there, I'm just going to run through the questions. I'm going to move fairly fast. Some of the questions I'll move through fairly quickly because they're relatively straightforward. Other questions are a bit more difficult and require me to slow down and give them a bit more attention. Really, the benefit of doing questions obviously is to cement the knowledge into your head about the topic, cement the basic facts into your head. But also, doing as wide a variety of questions as possible gives you familiarity with all the different styles and types of questions and exposes you to just about every angle with regards to types of questions. So when you come to do the actual exam, you have probably been exposed to just about every style of question. So it's good to do as many questions as possible, and this question booklet contains a fair range of questions, and it also contains some of the most difficult questions that have ever been on the photosynthesis topic. And it's important to do the most difficult questions. They're the ones that will make or break you. You know, everybody needs a grade A or a grade A or A star, or at least a grade B, and you can't get a grade B even if you aren't able to do the more difficult questions in the paper, or at least make a fairly good stab at them. So I would suggest that you do lots of past paper questions. Anyway, I'm just going to run through my question booklet here, which you should already have if you're doing tuition with me. If you're not doing tuition with me and you want help with tuition, there's my details on the screen there. You can get in touch with me by text. And I'll get you sorted out with a set of notes and a set of questions and so on. Okay, run through the questions then. Question 1 is asking, state precisely where the light dependent stage takes place in the chloroplast. Light dependent stage takes place on the surface of the thylakoids, or you could say grana. Thylakoids or grana. And why does it happen there? Because the thylakoids are the location of the photosystems. On the surface of the thylakoids are like flat plates, a stack of these flat plates making up a grana, and on the surface of each of these plates there's lots of pigment molecules, and that's where light energy is absorbed. With reference to events within the photosystems, explain the effects of increased light intensity. Well, the photosystem relies on absorbing light energy. But the energy is absorbed by all the pigments and then transferred to one primary pigment molecule. Well, if you have higher light intensity, which basically means more brighter light means there's more energy available. So if you say there's brighter light, then you just say more energy available, more energy absorbed by the pigments. The pigments are more, you could say, more excited. There's more transfer of energy to the primary pigment, and then if there's more transfer of energy to the primary pigment, there's more electrons than are excited or energized in the primary pigments. That'll be the first, somewhere in there, the first mark. And then the second mark was, if you're getting that happening, then you're going to get a faster release of electrons, faster emission of electrons, or more electrons emitted. All of those things would be okay. More electrons emitted, but really faster rate of release of electrons would be better. Explain the effect of different wavelengths of light on the activity of pigment molecules on the photosystem. Well, essentially that's asking about wavelengths of light, of course, colour of light, and it's asking how does the different colours of light affect a photosystem. Well, you know from the absorption spectra and action spectra absorption spectrum of the different pigments, you'll recognize that the pigments, all of them are pretty good at absorbing in the red end of the spectrum and the blue end of the spectrum, but not so good in the green end of the spectrum. So the first mark there might be something like the pigments in the photosystem absorb blue and red light very well, and you would see the different pigments then absorb slightly different colors, and they absorb different wavelengths or different colors. So the different absorption spectrum. Okay. By the way, you already have your answer scheme there in front of you. Anyway, so you can see essentially I'm just working from the answer scheme and I'm just elaborating on the answers. Part D of that question, it's asking what happens to the products of photolysis or photolysis, and I've given you 3 marks there. So you basically give 3 points for the 3 products of photolysis. And what are the products of photolysis? Well, one of the things that photolysis, basically the breakdown of water by light, and you're going to get electrons, you're going to get hydrogen ions, electrons, and hydrogen ions from the hydrogen atoms, and then you're going to get oxygen released. So the electrons that are released in photolysis, they're used to replace the 2 electrons that are they're used to replace the electrons that are lost from photosystem 2. So you're used to replace the electrons lost from photosystem 2. The hydrogen ions then get involved in a reaction with NADP, NADP and electrons to make reduced NADP or NADPH. And the oxygen is then essentially a waste product as far as the process of photosynthesis is concerned. It can be released as a waste product by the leaf, so it can seep out of the leaf into the atmosphere, or it can be used by the leaf in leaf cells in respiration. So those 2 answers would be good. So 3 products from photolysis, 3 uses for the 3 products. Describe precisely how the products of the light-dependent reaction are used in the light-independent reaction. And the key word there is precisely, so you want to underline that word precisely, because many students said the products of the light-dependent reactions are ATP and NADPH, and they said these 2 are needed to convert GP into TP, and they got 0 marks because they weren't precise enough. You need to precisely say what the NADPH does and what the ATP is for. And so what you would say is that the ATP provides the energy for the reaction where GP is converted into TP. It provides the energy, whereas NADPH reduces GP to TP or provides the reducing power for converting GP to TP. So it reduces GP to TP. Now you need to say those precisely, and the key word there was precisely. No, you didn't get the marks. Question 2 there is a question taken from an English board paper, and I chose it because it gave a good illustration of what a photosystem was. And it basically asks you then, it says, this represents a light harvesting system found on the surface of internal membranes of the chloroplast. Well, the internal membranes here would be the thylakoids, and the light harvesting system essentially would be the system of the photosystem. And so essentially they were looking 5 marks. Bear in mind it's an English board question, so slightly different mark scheme there. You can see in your picture, you can see the surface of the internal membrane. That would be, if you want to write underneath that, thylakoid, and then it shows those beads, those items there. Those are the pigment molecules, and you can see light energy being absorbed by a couple of the pigment molecules, and then it hops from pigment molecule to pigment molecule until it gets to the primary pigment molecule. So light energy has been absorbed by accessory pigment molecules and transferred to the primary pigment molecule. So essentially you get marks for saying that the photosystem, light harvesting system or photosystem contains various pigment molecules, and you can refer to them as accessory and primary pigment molecules. You can see light energy is absorbed by the pigment molecules. And you can say then that the light energy absorbed by the pigment molecules provides energy to the electrons in the pigments. Now, you mightn't have said that in a seance. You mightn't say that the electrons are excited or energized by the energy provided from the light. But you would certainly say that energy is passed from pigment molecule to pigment molecule. And often you would say that that's by resonance transfer. By resonance transfer. But you don't need that by resonance transfer because that wasn't given in last year's essay question, which is on photosynthesis, or 2019's essay question was in photosynthesis, and it didn't give the bi-resonance transfer mark, even though that was a term that used to be used to describe the movement of energy from pigment to pigment. And then you have to say energy is eventually transferred to the primary pigment molecule, which is a chlorophyll A molecule in the reaction center. And I would say all those things transfer to a chlorophyll A molecule, which is the primary pigment molecule in the reaction center. And then you would say something like that there's a range of different pigment molecules involved here in the photosystem, and each pigment molecule has a slightly different absorption spectrum of light energy, and the consequence of that is you can absorb as maximum amount of energy possible for the photosystem. Okay, so that's an English board question, but it's a useful one because it helps you to zone in on what a photosystem is. Now, this next question, and the question from a few years back, and it's quite difficult, and students find it quite difficult. But when you analyse it, it's relatively straightforward. And so it shows I think it's an algae. It's called Spirogera, and it's got a chloroplast inside, and the chloroplast is like a big long string of a chloroplast. It's called a spiral chloroplast, and it curves through the cell there, and you can see it. And you can see it running. It's called a spiral chloroplast. And you can see at some points the chloroplast touches the cell wall of the cell. And you see the experiment involved shining light onto the surface of the cell. And the light was a very narrow beam. In other words, the light's going just a very narrow beam. And you can see it's shown on to point A, and it's also shown on to point B. You'll see that point A is just the cell wall, and there's no chloroplast close by. Whereas at point B, on the second cell, you can see point B is directly above a chloroplast. And in that question there, I would almost suggest to the student, just for your own purpose, colour in the ribbon-like chloroplast. Colour it in green as spiral chloroplast so that you can clearly identify this is a very strange looking chloroplast, a spiral-shaped chloroplast. And then you can see down the bottom of the page, down the bottom of the first page, you can see then that the scientists introduced into the beaker with this algae, beaker of water with this algae, introduced bacteria that were very aerobic bacteria. And aerobic simply meant that they needed oxygen for the respiration. And also they said that they're motile. That simply means, that's another word for mobile, motile, just means they can move around, they can swim around these bacteria. And you can see what happened. There was a lot of bacteria congregated at point B, but not so many at point A. And it sort of said, then the first question was explain the difference in the number of bacteria found at point A and point B. And so, first of all, you'd see what the difference was. You'd see there's definitely more bacteria at point B. And if these bacteria are seeking out oxygen, it says up in the text of the question here, more bacteria at point B, that might be because there's more oxygen present at point B. There's more oxygen being released at point B. Okay, and why might that be the case? Well, the answer is that at point B, you can see the light beam is striking point B directly, and just directly under point B is the chloroplast. In other words, at point B, a beam of light will directly hit the chloroplast. So it'll be directly focused on the chloroplast, and that means you get maximum photosynthesis happening there. Whereas for A, if the light is shown at point A isn't directly on top of a chloroplast, and so you don't get so much oxygen. You don't get so much photosynthesis. So the answer again was more bacteria at B as there's more oxygen released at B. And the explanation would be as light strikes chloroplast directly at B, so photosynthesis is taking place at a greater rate. State a variable that would need to be controlled in this experiment. Well, you need the beams of light would need to be of the same colour, the same intensity, same brightness, same colour and same brightness essentially. So same wavelength, same brightness, same light intensity, brightness, light intensity. Investigation could be extended to look at the photosynthetic action spectrum. Well, that relates to how the rate of photosynthesis depends on the colour of the light being used. So you could use a range of different colours. So you just use the experiment B there, and you just shine each of the different colours onto that point B, and you could see how much photosynthesis is happening then for each of the colours. And you should find that you get the most photosynthesis at B if it's red and blue light, and not so much of its green light. And how would you detect there's a lot of photosynthesis at a particular point? The more bacteria. So you would say something like more photosynthesis, more oxygen released, or even more bacteria present for red and blue light, and not so many for green light. So you agree with me, once you get the hang of that question, it's not too bad, but it's quite difficult. You see it for the first time in the exam, and this is why you do these kind of questions. You do a lot of them as past paper questions, because you want to get yourself hardened and used to doing these kind of questions. You just want to get more confident that whatever question they throw at you in the exam, you should be able to cope with it, get the majority of the marks. Now, part C, once again, is a very difficult question. I think it's probably the most difficult question I've come across as a marker in marking exams. And students didn't do so well in it. But they didn't do so well because it was difficult. It wasn't totally unfamiliar, but it was difficult. The graph was quite difficult. So what do we see? We see photosynthesis can be investigated at a whole organism level. The graph shows the rate of carbon dioxide uptake in an evergreen tree in Northern Ireland over 24 hours in midsummer and midwinter. Okay, it's easiest here if you say cover up the bottom graph and just look at one graph for now, because it's a bit confusing. You try and look at both graph one, and just look at the top graph. And when you look at that graph, it should remind you of the graph you had at GCSE for the dawn dusk. Remember that graph where you're looking at both photosynthesis and respiration, and you had dawn and dusk, and you had the compensation points, and you remember then that at night time the plant was only doing respiration. Okay, so even those questions about photosynthesis, it brings in respiration, so just remember that. So at night time a plant would be doing respiration only, leaf cells would be only doing respiration, and then there will come a time in the morning dawn when the rate of respiration, rate of photosynthesis would be the same. And then during the daytime, the rate of photosynthesis will be much greater than the rate of respiration. And then you have dusk, where you also have another what's called compensation point, where the rate of photosynthesis and the rate of respiration are the same. And then go into darkness, where you only have respiration. So essentially, that's what's happening in this graph. Except that what's on the y-axis there is a bit awkward. The rate of carbon dioxide uptake. The rate of carbon dioxide uptake. Well. During the middle of the day, let's say there at 12 o'clock, you see on the top graph, 12 o'clock, during the middle of the day, what would the rate of carbon dioxide uptake by a plant? Well, it'll be quite high, because the plant will be doing lots of photosynthesis. So it'll be taking up a lot of carbon dioxide. But bear in mind, the plant would also be producing carbon dioxide in respiration. But in the middle of the day, it's going to be using up more carbon dioxide than it's producing, because photosynthesis is much faster than respiration. So you can see there a very positive value for the rate of carbon dioxide uptake in the middle of the day. And then at night time, you see that the rate of carbon dioxide, so for example, from 0 to say 6 o'clock in the morning, 0 to 6 o'clock, see the rate of carbon dioxide uptake is negative. Our rate of uptake is negative. Well, that means if you've got a negative rate of uptake, that means output, that means carbon dioxide is being released. Yeah, so that's what that suggests. So it then goes over, and part one says, the graph shows information on the rate of net photosynthesis. And what they meant by net photosynthesis was the overall rate of photosynthesis when you subtracted from photosynthesis, you subtracted the effect of respiration. When you subtracted the effect of respiration. Whereas rather than gross photosynthesis would be just where you're just considering photosynthesis. If you're just considering photosynthesis, then the graph would show zero uptake during the night time, and zero uptake during the night time, and positive uptake during the day. But the fact that it goes negative, this graph, tells you that it can't be just photosynthesis, that respiration must be considered as well. So this is a difficult part, and most students find this difficult. So what you would say here, what's the evidence that this is net photosynthesis? In other words, photosynthesis taking account of respiration, and you'd say, oh, there are negative values of rate of carbon dioxide uptake. There are negative values of carbon dioxide uptake. And this then relates to the fact that respiration is being taken into account, that this graph is taking into account respiration as well. So it's not just about photosynthesis and respiration put together. Okay, so that was difficult to get past that. Now, the rest of the question then was about comparing the two graphs, midsummer and midwinter. And just looking at the graphs, then you can see that the bump in the middle of the two graphs in the middle of the day, that bump is much bigger in midsummer than it is in midwinter. And you, first of all, you had to describe. You'll notice in the question it says describe and explain. So if we describe, first of all, we could say in midsummer carbon dioxide uptake is much higher. Carbon dioxide uptake is much higher in the middle of the day, and there is a net uptake over a longer period. So to get that described, Mark, was difficult. First of all, you had to say the bump was higher. You couldn't say the bump was higher. Of course, you had to say the carbon dioxide uptake was higher during the middle of the day, and also that the carbon dioxide uptake was positive for a longer time. So you'll see in midsummer that the carbon dioxide uptake was positive from 0600, that's 6 in the morning, until 6 in the evening, 1800. So for a long period of time. Whereas in midwinter, it was only positive from about 9 o'clock in the morning to about 3 o'clock. So to get that described, Mark, there, you needed both those. You had to say it's much higher, the carbon dioxide uptake is much higher during the day, and there's a net uptake over a longer period of time. Another thing you might notice is that the carbon dioxide output is higher in midsummer. Sorry, carbon dioxide output. So we're now talking about during the night time. It's higher in midsummer. In other words, the line extends down more below the axis. In midsummer, it goes down as far as -2. In midsummer, that's the carbon dioxide output. Whereas in midwinter, it went down to -1. So this question is particularly difficult. You got 2 marks for describe. There are 2 marks, and you had to describe 2 aspects of it, and then you had to come up with some explanation. So first of all, why is the bump? Why is the carbon dioxide uptake higher in the middle of the day in summer? Well, that's getting down to the fact that you've got a faster rate of photosynthesis. And why would you have a faster rate of photosynthesis in the summer? And you'd say higher light intensity, and it's warmer in the summer. So both those would contribute to a higher rate of carbon dioxide uptake in the summer. Photosynthesis is faster in summer temperatures. And then what about the fact that the carbon dioxide output, in other words, the distance the line goes below the axis, in midsummer is greater. You could see respiration rates are greater in midsummer due to the fact that you had higher temperatures. So there you are, there's your 4 marks. You can check them out on the mark scheme there, but they're particularly difficult. But the good thing is exposing yourself to that question. That's on the course discussion about photosynthesis and respiration at different times of the day. Now, the thing with this question is that there's a bit of an overlap between photosynthesis. This question also appears later on in the ecosystems topic. And so maybe if you've only done photosynthesis at this stage and you haven't done any of the ecology or systems topics, then it's maybe not just as easy for you. And once you would have completed the ecosystem topic where this is also dealt with, where we look at something called net primary productivity, then this would make more sense to you. So bear in mind this question was at the end of the year when you've done the whole of Module 5. Okay, on we go. On to question 4. Essentially question 4, when we look at that question, is really a repeat of question 1. I think it's essentially the same question as question 1. And it points up the fact here that question 4, I think it's about 8 years old, that question. So back about 2012. And the question on the front page, question 1, is much more recent. So you can see how the older question is basically used again to create the newer questions. So this is the other value of doing lots of past paper questions. You come across questions that are simply replicated in later papers. And so you just make yourself as familiar as possible with all possible questions. So where position of chloroplast photosystem 1, photosystem 2 located? Answer: on the thylakoids or grana. And then explain the effect of increased light intensity. That's the same question as question 1. Now, increased light intensity means more light energy, means brighter, means more light energy absorbed by the pigments, and therefore electrons would be emitted or rejected at a faster rate by the photosystem. Different wavelengths and the activity of pigment molecules. You'd say different pigment molecules have absorb different wavelengths and different colors, which is their absorption spectrums. And then you would see some maybe add in that blue and red pigments are blue and red wavelengths are more strongly absorbed than green. Green's reflected. During this process, during the process of photosynthesis, water molecules cause to yield electrons. This is an older version of this question about the photolysis. You sort of see the language that's used there. Used to yield electrons and hydrogen ions. What happens to the electrons from photolysis? They return to photosystem 2 to replace the electrons lost in photosystem 2. What happens to the hydrogen ions? Hydrogen ions are used along with electrons to create hydrogen to combine with NADPH. Combine with NADP to make NADPH. State the name of the compound X on the diagram opposite. Compound X then in the non-reduced form will be NADP. And what's the role of the reduced form of that, which is NADPH? It's to reduce glycerate phosphate to triose phosphate. So it's acts as a reducing agent in the reaction, glycerate phosphate to triose phosphate. Many turns the Calvin cycle. We're coming on to the Calvin cycle and other questions here. The answer is 6, and we'll discuss that later. In the form which glucose is stored within the chloroplast, that's starch. You could also say, if you remember back to your AS, that could be amylose or amylopectin. Would get that as well, but starch is what they're after. Now, question 7 is quite an old question. It's about 10-year-old, and it shows something. It says it's supposed to represent a photosystem, and suggest a photosystem looks like a funnel. Well, it's not really suggesting it looks like a funnel. What it's suggesting is that the way in which the photosystem works is it's like a process of funneling. And this is where the word antenna complex comes from, this idea that the photosystem is like a satellite dish or an aerial that collects. In this case, the photosystem is collecting light energy, and it's funneling it all this is funneling the light energy through all the accessory pigment molecules to one single pigment molecule in the reaction center there. And if you want to write on the reaction center, chlorophyll A molecule. Primary pigment chlorophyll A molecule. And the reason I say that is that last year, 2019, or the year before, 2019, the essay was in photosynthesis. And if you look at that essay, you'll see that you specifically had to say the primary pigment is a chlorophyll A molecule. And if you didn't say that, you missed out the mark. Where the chloroplast photosystem is found, that's thylakoids or grana. 2 types of pigment molecules. This is a very old question. 2 types of pigment molecules. So you could say chlorophyll molecules, chlorophyll A and chlorophyll B, xanthophyll, carotene, those sorts of things. It's 2 types of pigment molecules. I think if you said chlorophyll A and chlorophyll B, you mightn't get that. You're better to say chlorophyll A, chlorophyll B, and put another one, xanthophyll or carotene there. The advantage of the light harvesting process has been absorbed by many different types of pigment molecules. Many pigment molecules means more light energy absorbed for photosynthesis, because the different pigment molecules absorb different wavelengths. What happens as a result of transfer of energy to the reaction center? Electrons are emitted. Light harvesting produces ATP, NADPH, and oxygen. And you have to say what each of these 3 do. ATP provides energy for the conversion of GP to TP. NADPH reduces GP to TP. By the way, you can get away with saying GP to TP rather than glycerate phosphate to triose phosphate. So far, anyway. Personally, I would say I would use their proper names. I would say glycerate phosphate to triose phosphate rather than GP to TP. And oxygen. What happens to the oxygen forming? You'd say released from the plant as a waste product or used in respiration. How are chloroplasts structurally adapted for photosynthesis? Well, this is a kind of a synoptic question going back to AS. I would, for photosynthesis 4 mark, it's a made up question by myself just to get you to zone in on chloroplast. I would say chloroplast contains internal membranes called thylakoids, which are grouped together in grana. Their purpose is to create a large surface area of surface on which to position chlorophyll and other pigment molecules in photosystems. And the idea of the large surface area is to maximize the area available for pigment molecules to be located so that they can maximally absorb light energy, so they're completely exposed to light energy. So if you have your thylakoid sort of looking like a sort of a plate, and essentially on the surface of it you've got your pigment molecules sitting on the surface of the thylakoid. Well, if you have a lot of thylakoids and a lot of surface area, then you have room for all these pigment molecules. You have a large surface area for pigment molecules positioned on the surface of the surface of the surface of the plate, surface of the thylakoid plate, and therefore you can get maximum absorption of light energy. I think that's probably as much as you would want to say. You could mention the grana then. The grana is the location of the reactants and reactions of the light independent reactors. Over the page to question 9, which is your Kelvin cycle. Kelvin cycle is not that difficult to learn, and I would definitely recommend drawing it out. And you'll see in this question specifically looking at the number of carbons present in the different molecules. Different molecules here. Up the top there you see ribulose bisphosphate combining with carbon dioxide to make 2 glycerate phosphate molecules, and that step where it combines with carbon dioxide is carbon fixation, and they're fond of asking about carbon fixation. Complete the diagram to indicate the number of carbon atoms. So ribulose bisphosphate has 5 carbons and glycerate phosphate has 3 carbons. And describe the different roles of ATP in the cycle. Well, first of all, the different roles of ATP, and notice it's plural, roles of ATP. And if you look in my notes there, you'll see that ATP is needed in 2 places in the Kelvin cycle. First of all, it's needed to provide energy for the conversion of glycerate phosphate to triphosphate, so it provides energy for that step. And then if you look at a step where you go from ribulose phosphate to ribulose bisphosphate, you'll see that ATP is needed there as well. And ATP is needed there for 2 purposes. You'll see that when the ATP is used in the conversion of ribulose phosphate to ribulose bisphosphate, you'll see that its ATP creates ADP, and a phosphate disappears. Well, that phosphate is needed to convert ribulose phosphate, which is just 1 phosphate, to ribulose bisphosphate, which means 2 phosphates. So that phosphate is needed to convert the ribulose phosphate to ribulose bisphosphate. Now, when I was marking this question, we had to also know that not only did the ATP provide the phosphate for the conversion of ribulose phosphate to ribulose bisphosphate, that the ATP also provided energy in that step as well. And so I'm just looking at the original mark scheme here to see what they included in the original mark scheme. They said just provides phosphate for the conversion of triose phosphate to ribulose bisphosphate, or ribulose phosphate to ribulose bisphosphate. So even though we had to, in the exam, we didn't give you the mark if you didn't say that ATP was needed to convert and provide phosphate to convert ribulose phosphate to ribulose bisphosphate. You also had to say providing the energy, but in the actual mark scheme they left out the energy bit. So there you are. Part B, particularly difficult, especially you hadn't seen something like this before, but it's good to look at it now and be familiar with it. What you're seeing is they're looking at a leaf from top to bottom, and they're measuring, as you move down from the top of the leaf to the bottom of the leaf, they're measuring the light intensity, or the light that's available inside the leaf. And that is, I think that's line Z, isn't it? Z is the amount of light available. So you can see near the top of the leaf, Z is high. The amount of light available, and as you go down through the leaf you get less and less light. That sort of makes sense, If the light the light shining down on the top of the leaf, then as the light moves down through the leaf, there'll be less and less of it available as it gets absorbed by the layers near the top. And then the other thing they're measuring is the amount of carbon dioxide fixed by RUBP. The amount of carbon dioxide fixed by RUBP, that sounds like a difficult thing to get your head around, but really that's really just a measure of the rate of photosynthesis. It's another way of saying the rate of photosynthesis, because if there's more carbon dioxide fixed by ribulose bisphosphate or involved in carbon fixation, then the rate of photosynthesis is faster. Okay, and what that graph's showing you is at the very top of the leaf, there seems to be no carbon dioxide fixed. And then as you move down into the palisade layer, you see that the amount of carbon dioxide fixed shoots away up enormously. And in fact, it looks like most carbon dioxide is fixed in the palisade layer. And then you get less carbon dioxide being fixed as you move down through the spongy mesophyll layer, right down to the bottom of the leaf, where it would appear in the lower epidermis you have no carbon dioxide fixed. So if you just convert the idea of carbon dioxide fixed there into photosynthesis, just think of it as photosynthesis, then you see that there's no photosynthesis in the upper epidermal layer, lots of photosynthesis in the palisade layer, quite a bit of photosynthesis in the spongy mesophyll layer, but less in the palisade layer, and no photosynthesis in the lower epidermis layer. So once you've figured that carbon dioxide fixed essentially equated to photosynthesis, then the question becomes a lot more manageable. So suggest a reason for the steep reduction in light intensity at point Z. Point Z is at the top of the leaf. They're looking for a steep why is there a steep reduction in light intensity? That was difficult. About 50% of students got that. 50% of students said light gets reflected off the waxy cuticle. So a lot of the light that strikes a leaf gets reflected. And this really comes from another topic too. This comes from an ecosystems topic where this is discussed. Energy flow through a leaf is discussed. So having some exposure to this other ecosystems topic would help you here maybe as well. So the answer to that is a reflection of light at the waxy cuticle, or just the cuticle. Explain the pattern of carbon dioxide fixation through the leaf section from X to Y. In other words, from the top of the leaf to the bottom of the leaf. So explain the pattern. So in each case you'd have to say whether there was any carbon fixation or not. So no carbon fixation in either the upper epidermis or the lower epidermis. So there's no carbon fixation there in the upper epidermis or lower epidermis. And you would say, why is there no photosynthesis? Why is there no carbon fixed? No photosynthesis in the upper or lower epidermis? And the answer is no chloroplast. So that was your mark. So there's no carbon fixation in either epidermal layer, upper or lower epidermis. There's no chloroplast. And then the next thing you would maybe go at is there's a lot of carbon fixation in the palisade layer. So high rate of photosynthesis in the palisade layer. And for the GCSE, you would know the answer. Why is there a high rate of photosynthesis in the palisade layer? You see, there's maximum number of chloroplasts. That's where most of the chloroplasts are in the palisade layer. There's a second factor that would affect the amount of photosynthesis or carbon fixation in the palisade layer as compared to the spongy mesophyll layer, where you can see in the spongy mesophyll layer there's less carbon fixation, less photosynthesis. Now, why would you have less photosynthesis in the spongy mesophyll layer than in the palisade layer? Two reasons. First, the spongy mesophyll layer doesn't have as many chloroplasts, and secondly, it's further down the leaf and the light intensity is lower. So less light's reaching the spongy mesophyll layer. So let me just summarize the mark scheme there for you. So first of all, you're saying there's no carbon fixation in either epidermal layer because there's no chloroplasts available. Then you could say there's maximum fixation in the palisade layer as there's high light intensity in the palisade layer, and also the highest number of chloroplasts in the palisade layer. And you could say then about the that would get you the 3 marks. Alternatively, you could say there's less carbon fixation in the spongy mesophyll layer for 2 reasons. There's fewer chloroplasts and there's less light reaching the spongy mesophyll layer. So there you are, a scary looking question. But if you convert the idea of carbon dioxide fixed by RUBP, if you just translate that into photosynthesis, then a GCSE student could understand this question. It just shows the difference in that statement. That statement confused a lot of students because they just didn't figure what it meant. Okay, on to question 10. Glycerate phosphate is produced during the light independent stage. Um, why is the light independent stage sometimes called the dark stage? Explain why the light independent is more appropriate term. Well, this is getting at the idea that you can't really refer to the light independent stage as as a stage that doesn't need light, because you actually need light beforehand to get NADP, H, and ATP, which are needed then for the light independent stage. So the fact that the light independent stage actually requires ATP and NADPH, which are made in the light dependent stage. Precise location of the light independent state, that's the stroma. And then and this is an interesting question that has come up several times in the past 20 years or so. Explain the changes in concentration of triose phosphate when light is no longer available as shown in the graph below. Well, you see when light is available, the levels of triose phosphate and glycerate phosphate are relatively constant and at a sort of middling value. But as soon as it goes dark, the amount of glycerate phosphate starts to increase and the amount of triose phosphate decreases. Well, why would the amount of glycerate phosphate in the Calvin cycle start to increase when it goes dark? Because when it's dark, you're no longer getting ATP and NADPH being produced. And if you no longer get NADPH and ATP produced, then you can't get glycerate phosphate converted into triose phosphate. And so the glycerate phosphate in the cycle, it starts to build up, because it's not been converted. And the triose phosphate that has been produced, it gets used up to make glucose or ribulose phosphate. So if you understand that, then so it's like a stop in the production line. If you don't get ATP and NADPH, then you can't get that step that converts glycerate phosphate to triose phosphate. So any glycerate phosphate that is made, it just hangs about, it's just there, and it accumulates, it builds up. That's what the build-up. And the triose phosphate, it keeps getting turned into glucose, so it diminishes, the amount of it decreases. Absorption spectra. Use the information provided. Plant leaves are normally green in colour. Well, that's almost like a physics question. Why is something a particular colour? And the answer is it has absorbed all the colours of light in the spectrum, but it must have reflected green. So that's why are leaves green? We should say leaves reflect green because they don't have pigments that can absorb green. Green is not well absorbed, whereas all the other pigments are absorbed. Actually, the full answer to that question for 2 marks was green is not well absorbed, or green is least absorbed, and therefore it is reflected. And that's what makes the leaves green. So whatever is reflected off the leaf into your eye gives you the colour of the leaf. Okay. An action spectrum. What's going to be an action spectrum? Well, that's that's a measure of the rate of photosynthesis for different colours. So you just use a range of different colours and measure the rate of photosynthesis. So how photosynthesis rate varies with wavelength. Is good. Wavelength is colour. On to part B here. Now, in part 2, students didn't like, part 1, B part 1. Students didn't like B part 1 because this is not strictly speaking on the specification. It doesn't ask you in the specification to know about an experiment to measure the rate of photosynthesis. However, they might expect you to go back to GCSE and remember how to measure the rate of photosynthesis from GCSE. And remember in GCSE you used a upside down funnel in a beaker of water. You'll remember this, and then you had like a test tube over the top here. Then you had, this was full of water. This is only water in here. And then you had your wee plant in here, which was your wee Aladea plant. Then your wee Aladea plant, you shone light on the whole arrangement here. Shine on some light. Here's a big lump. You shine light onto the plant like that. And what happens is that the wee plant starts to produce bubbles of oxygen. Okay, and the bubbles of oxygen then collect up, and they can collect up in the test tube up here at the top. Bubbles go right up. Here we go. The bubbles of oxygen are collected, and in the top of the test tube. And of course, instead of the test tube, you could use a measuring cylinder, and you could shine light on the plant then. And you remember this from GCSE, so this is a way of measuring the rate of photosynthesis under different conditions. So even though this wasn't on the A-level course, the examiners expect you to come up with some sort of suggestion of this kind of experiment. So they want you to say something like, use a water plant, such as Elodea, Canadian pondweed it's called, Elodea, and you collect the oxygen produced by the water plant and measure the rate at which oxygen is produced, and that will give you a measure of the rate of photosynthesis. And of course then you could use that, you can use that setup then to change all those factors up in the graph above. So you change the light intensity, so you just, if you remember GCSE, you can move the lamp further back or closer and change the light intensity and then measure the rate of photosynthesis rates for the different light intensities. Other things you've changed in that experiment are temperature. So effectively you could change the temperature of the water. That would work around the plant. And you could change the carbon dioxide level. That'll be a wee bit more difficult to measure change the carbon dioxide levels, but essentially you could add something to the water that would introduce more or less carbon dioxide to the plant. So use your knowledge of the light dependent reactions. Explain why the rate of photosynthesis is higher B than a day. So you have to talk to the light dependent reactions. Why is the rate of photosynthesis higher B than a day? Well, if we look at B, we'll see that the difference between B and A is B's got a higher light intensity. A's got a much lower light intensity, so B's a higher light intensity. So essentially it's just saying why is the rate of photosynthesis higher with a higher light intensity? And so once again, it's the same old question. You just say if there's more light available, higher light intensity, more light energy available, more light energy absorbed by pigment molecules, more light energy transferred to the primary pigment, and a faster rate of release of electrons, and then faster production of ATP and ADPH, and faster production of glucose. There's a lot you could write there. But if you said essentially higher light intensity, more electrons emitted per second, or a faster rate of electron emission, and therefore faster production of ATP and ADPH for the light independent reactions. Use your knowledge of the light independent reactions. Explain why the rate of photosynthesis is higher at C than at B. Well, what's different about C than B? Well, you'll see that at C there, you get the same light intensity, because they're both directly above each other on the graph there, B and C. But the difference is C's got much higher CO2 levels and C's got a higher temperature. So really, this is why this has got 4 marks. It's basically saying why do you have a higher rate of photosynthesis when you have higher CO2 levels and higher temperature for the light independent reactions. And as you take each of those in turn, you'd say higher CO2 levels means more CO2 available to combine with ribulose bisphosphate to make glycerate phosphate. And then you'd say higher temperature means a faster rate of enzyme action. And then you mentioned that Ribisco is the enzyme required for the fixation of carbon dioxide. So there you are. So the 4 marks are higher CO2 levels means more CO2 combined with ribulose bisphosphate, and that therefore makes more glycerate phosphate, more glucose, etc. Higher temperature means enzyme action is faster, and then any reference to Ribisco. Ribisco is an enzyme used there in the reaction. And on we come then to section B there, question 11. And question 11 is 2019 question. That's quite a nice question. Students, there's 12 marks there. Most students got all 12 marks for the first part, which is describe and explain how plants use light energy to make glucose. Very short and sharp little line. Describe and explain how plants use light energy to make glucose. And the second part of it, part B, was worth 6 marks. Students didn't do so well at it. So let's just talk with it. You've got the mark scheme yourself there anyway, so you can check through the mark scheme. But essentially, you're going through the light dependent reactions and then through the light independent reactions, and you just I think it's 12 marks you're looking for there. You get 12 marks. It's actually quite easy to get 12 marks. So if you look at the mark scheme there, you'll see the points that they give marks for. They said the light dependent stage, light is harvested over a range of wavelengths. An alternative that they allowed there was light is harvested by a number of different pigments. So that is an alternative to that. So if you just said light's harvested by different pigments, if you said this light harvesting happens in the thylakoids or grana, that was good. And then at some point in your answer, the whole question you had to mention there was a photosystem 2, and then later on there's a photosystem 1. Then notice very clearly point 4 there, chlorophyll A molecules become excited. In other words, you had to very clearly say there's a chlorophyll A molecule in the reaction center. The primary pigment is a chlorophyll A molecule. You couldn't just say a chlorophyll molecule. And then there's point 5 then goes on to talk about photolysis, and it says that the electrons lost from photosystem 2 are replaced from photolysis. And then think about the electrons pass through a series of carriers in the electron carrier system, and at certain stages in the electron carrier system there's sufficient energy to make ATP. Or you could say that this is called photophosphorylation. So essentially not a wild lot of marks there for describing the electron carrier system. Although there are other, if you had my notes there, you would see that there's a number of other questions that I have identified from other years where they ask questions about asking you to compare respiration and photosynthesis in terms of their electron carrier systems. And there's a number of other questions. I think that's worthy of having a good look at the electron carrier system. There's a lot of important points there, and if you check my notes there, you'll see that I've kind of gone into in some detail. So they give a very limited number of marks there for up to that point, 6 marks really to get you up to the point of the electron carrier system. Sorry, 7 marks. And then it's basically saying that the electrons that pass through the electron carrier system, whose energy is extracted and used to make ATP, these electrons then go into photosystem 1 to replace the electrons that are lost from it. And the electrons that are lost from photosystem 1 combine with hydrogen ions from photolysis, and then those hydrogen ions create hydrogen, which then combines with NADP to make NADPH. And then at point 12 then says ATP and NADPH are used in the light independent reactions. So after point 12 then you're into the light independent reactions. Point 13 says the light independent reactions happen in the stroma, which is easy enough, and it tells you that carbon dioxide combines with ribulose bisphosphate to create 2 glycerate phosphate molecules, and mark then for saying that reaction is catalyzed by the enzyme Ribisco. And then it's saying then that NADPH is used to reduce GP to TP, and ATP is needed also in that reaction step to convert GP to TP. And then it goes on to the last point there. It's saying that 1/6 of the TP molecules, triphosphate molecules, are used to make glucose, and the other 5/6 are used to regenerate ribulose bisphosphate, or some words to that effect for that last point. And so there you are. That's there's 18 points there, and you had to get 12 to get the full 12 marks there. And the second part was much more difficult. It says, with reference to the factors that limit the rate of photosynthesis. With reference to the factors that limit the rate of photosynthesis. What are the factors that limit the rate of photosynthesis? From GCSE, there was light intensity, temperature, and CO2 levels. So you have to refer to those. It then says, suggest why leaves in shady positions. Leaves in shade. So leaves that would be covered over by other parts of the plant and wouldn't be getting sufficient light. Leaves in shady positions are often broad, thin, and darker. So there's 3 things. Broad, why are leaves in shady positions darker, broader, and thinner? So why are they darker? Why are they broader? Well, broader because if you're a shaded leaf, then it's just going to be broader because you have a larger surface area, and therefore you can have more. Larger surface area means you can absorb more light energy. So where there's limited light energy on the forest floor with shady plants, they tend to be bigger, broader leaves to absorb more light energy. Thinner essentially means that the leaf is thin from top to bottom, and there's a couple of points there you could say. If the leaf's thinner, then light's able to penetrate down through all the layers in the leaf and reach all the cells. Equally, you could also say if the leaf is thinner, there's a shorter distance from the bottom of the leaf to the top. That means there's a shorter distance for carbon dioxide to diffuse from the stomata on the bottom up to all the layers of the leaf, all the layers in the leaf, and darker in colour. Why are leaves in shady positions darker in colour? Well, darker here is getting down to the fact that they're darker green. If they're dark, so if they're darker green, that means they're more chlorophyll, and therefore they can absorb more light energy. So that was the mark for that. And the last point there, it says, what about the leaves near the top of the canopy tend to have a greater stomatal density? That was the hardest point. What is it about the leaves at the top of the canopy that means they might benefit from having greater stomatal density, in other words, having more stomata? And the answer there was that at the top of the canopy, this is up the top of the trees in the forest, for example, there would be plenty of light and adequate temperatures, but the factor that may be in short supply would be carbon dioxide. Carbon dioxide may be in short supply because the other two factors are in plentiful supply. And if carbon dioxide is in short supply, then having more stomata to allow more carbon dioxide to enter the leaf then might be an advantage. So that was quite a difficult point. So there you are. That's a quick run through those photosynthesis questions. And as I say, exposure shift has a wider range of photosynthesis questions, all sorts of questions, and is a good thing because it just makes you more confident. It just makes you more more knowledgeable about all the different types of questions. And the chances are that when you come to your exam, one of the questions you get, you're going to hit on some of the questions you've done before, because there are only so many questions and they tend to just be recycled in one form or the other. So that's good then. If you felt, if you don't have, if you're seeing this video and you don't have, you're not doing tuition with me, and that means you don't have access to all these materials, and I assure you that there's a tremendous amount of materials here, and the notes are comprehensive and contain a lot more questions and a lot more areas that are covered in other questions from a long time ago. Then get in touch with me, and I can get you a full set of all the materials, and then you'll benefit more from these sessions. Okay, thank you very much. My name's Dan. All the best. Bye.